Is Stefan Stoll again. He's going to give another lecture, and he's going to tell us this time about Pulse EPR spectroscopy. Right after this, we should have a second talk given by Dennis Proshlyakov. Is Dennis here? Not yet. Which you may not know is Dennis was, of course, at the meeting together with his son Yegor. Yegor, which you might not know, is actually a high school student, and they just had his high school graduation where they had to fly back and zip back in. So, if Dennis doesn't show up, we have a few options who are ready to stand in. I see one of the options laughing and hiding. And we hope that maybe Dennis will show up. And until then, Stefan will tell us about Pulse EPR.
So, good morning. Yesterday, I showed you a brief illustration of what continuous wave EPR can do for you in terms of learning about structure of an active site in a metalloprotein. Today, we're going to talk about a different technique called Pulse EPR spectroscopy that yields similar information, although at a higher resolution. So, the goal today is going to be just you should learn what you can learn from Pulse EPR. The goal here is not to actually make you understand Pulse EPR in detail. If you go to the Dropbox account and go to the 2014 folder, there is a slide deck with about 60 slides from 2 years ago on Pulse EPR that contained a lot of spin physics. So for those who like to look at equations and understand what's going on deeply, they should go there. Today I'm going to give a more introductory overview and just walk through a bunch of examples to illustrate how you can use the technique. Okay, so the content is going to be first some very basics about Pulse EPR. Then second, we're going to talk about magnetic nuclei and their interactions. Now you might find that strange because EPR is about electrons. But Pulse EPR is actually mostly about nuclei. It's a way to measure NMR via the electron spin. And then third, we're going to go through some examples from the literature. Old literature and very recent literature. Okay, so first the Pulse EPR basics. And if you're trying to find the slide deck on the Dropbox folder, I will upload it after the lecture. Okay? So EPR spectroscopy looks at spins and their coupling. Now, you might be familiar with these pictures, of course. They come from crystallography. They're derived from electron density, or spatial density, and we draw these cartoons of the location of certain nuclei. This is the protein we're looking at in a Pulse EPR practical. It's myoglobin with a hydroxy group. It's a ferric iron, low spin, spin 1/2. Now, so this is the crystallography view. The EPR spectroscopy view is this. It's a network of coupled spins. At the center of our attention is the strongest magnetic particle, which is the electron. And it sits in an environment of other magnetic particles, which are the nuclei, protons, and nitrogen-14s. And since both the electron and these nuclei are magnetic, they feel each other. They interact. And using Pulse EPR, we're going to measure these interactions, and not just around the electron at the metal ion, but also on the ligands. So if you will, you could say Pulse EPR is ligand EPR, right? CW EPR is metal ion EPR. All right. So here is a little overview. If you look myoglobin with CW EPR, we can understand things about the metal center, and maybe we can see a ligand nucleus. Mostly we just see the metal center and the hyperfine coupling from the metal ion, like copper or cobalt or manganese, what I've shown you yesterday. So CW EPR is a very limited field of view. Now with Pulse EPR, we can look further away. We can look at magnetic nuclei that are further from the electron spin, like protons, nitrogen-14s, and carbon-13s. We can go into the first or even the second ligand sphere. We can look at hydrogen bonds. And we could also measure the distance to other electron spins. So if you have a protein with two metallic active sites that are paramagnetic, you could measure the distance between those. And Pulse EPR can capture magnetic nuclei within a few angstroms of the electron spin, and you can see electron spins, other electron spins, within a few nanometers, so about tenths of angstroms, up to like 60 angstroms. That's how far you can look with Pulse EPR. So it's the same principle. We're looking at EPR spins, just we're using a completely different instrumentation to get more information about what's more remotely away. So let's review from last time what EPR samples look like. Just like CW EPR, Pulse EPR is a solid-state spectroscopy. So we always freeze the sample. So we have typically frozen aqueous solutions of proteins. Here's our cartoon from yesterday about the sample. We put it in a magnetic field, and all the different proteins have random, uniform distributions. So we call this a powder, right? It's a random uniform distribution of static orientation of the molecules. They don't tumble, and they don't diffuse left and right and up and down. So it's frozen and rigid. Now, yesterday we also saw that a CW EPR powder spectrum is recorded as a first derivative, and what you should do mentally always, picture the integral. And we saw that this is the actual, when you integrate this, you get the absorption spectrum, and we saw that it's really broad. And the reason that it's broad is that different proteins in the sample have different resonance fields. If you measure them all together, you get a broad spectrum. Now, there are three principal reasons of why proteins have different resonance fields. The first one is that some of the magnetic properties are anisotropic, which means you take a protein, look at its resonance peak. If you turn the protein a little bit, the resonance peak will move. Okay, we talked about that yesterday. The second origin of these broadenings is that there's structural heterogeneity sometimes. Sometimes, one protein might be a little squeezed along the axial direction of the complex, or another one might be slightly stretched. This leads also to slight difference in the resonance fields, and that leads to broadening as well. And the third one is unresolved splittings. A reason for broadening is a hyperfine coupling to magnetic nuclei and a coupling to other electron spins. And that is now the information that can be extremely valuable. It's hidden structure in the spectrum, and it's also hidden information about the structure of the protein. And with Pulse EPR, we can tease this unresolved structure, which is shown here, out. Here you see the powder spectrum. And if we had just a single protein, a single orientation, if all the protein molecules were aligned, orientation-wise, we would get a very nice resolved spectrum, as if you had a protein crystal. Okay? Now, we don't have that. We have a frozen powder, but Pulse EPR can actually help us tease out these really small splittings and couplings that tell us about other magnetic nuclei. So Pulse EPR is very different experimentally from CW EPR, even though conceptually it's not super different in terms of what it gives you. So I'm going to walk briefly through what the main differences are. In CW EPR, we continuously excite the spins with low microwave power, and we measure the absorption, so it's an absorption spectroscopy. We measure the steady state response, right, because with CW, and it's, of course, low resolution, as we've seen. Pulse EPR, in contrast, does not use CW excitation, but pulse excitation. Very short, intense pulses with powers of watts to kilowatts. It is an emission spectroscopy, because what we are measuring in Pulse EPR is a transient response after the excitation. So we're not measuring a weak absorption while we're illuminating it, like in a UV vis spectrophotometer or CW EPR, but we're like hitting it with a hammer, hitting the spins with a hammer, waiting a few nanoseconds and see if they respond back. And this will give us high resolution. So the most important thing, if you haven't done NMR or know too much about NMR, is to realize that Pulse EPR is something very, very different from optical absorption. Right? In optical absorption, typically we just have a ground state and an excited state. If the radiation gets absorbed, the system goes from ground state to the excited state and just absorbs the energy. Now, EPR is just like NMR, coherent spectroscopy, which means things are a little more refined. So imagine here, you have an external magnetic field in a spectrometer, and this is a picture of the electron. You can picture it as a spherical bar magnet that's spinning around its axis, just like the Earth. And the arrow, so the yellow arrow indicates the spinning motion, and the black arrow indicates the magnetic moment, or the spin, if you will. Now, if we apply microwave to this for a specific amount of time, which we have to determine, nanoseconds, and a specific amount of power, we can flip the spin. We can go from spin up to spin down. And in an energy level diagram, this would correspond to this. We have a ground state. First, the spin is in the ground state. After applying what this is called a 180-degree pulse, the spin is in the excited state. Okay? With this, we can't do anything. We couldn't do anything. Now, we can just switch this power off halfway through. And if you switch this power halfway through, off, we end up with the spin looking sideways, right? And then the spin sideways state is not something you've probably heard about. You just know alpha or beta, spin up or spin down. But spin sideways is a special state. It is, you can actually picture it like this. So people draw it like this when they draw an energy level diagram. It's not the ground state. It's not the excited state. It's a superposition of the two. The spin points sideways, and the characteristics of this state is that it's non-stationary. It precesses. So the spin precesses. If it's spin up, it just sits there. If it's down, it just sits there. But if it's sideways, it precesses in the magnetic field. And that frequency of the precession is what we're going to measure. That will tell us about the environment of the electron spin. Okay, so that's important to realize. So Pulse EPR uses pulses of short pulse of microwave to manipulate these spins. Some people call it spin gymnastics. Okay, and here's how it works. This is a very basic sketch of a Pulse EPR spectrometer. You have a source of microwave, then have a little device that actually makes your pulse. Like switches the microwave on and then switches off, kind of like a shutter. That's really fast on a nanosecond time scale. Then this is all low power. Then it goes to a high power amplifier, which is always very expensive. Let's go back here. And then the red arrow indicates now we have high power pulsed microwaves. They go to the resonator in the magnetic field and the EPR spectrometer and excite the spins. Now, after the pulses are over, we can switch on the detector. And if you look here at the black arrows, that's a weak response from the sample. There's a little shutter in front of the receiver to make sure we don't kill it when we hit it with the sample with pulses. But once the pulses are over, a weak emission comes from the spins, gets amplified, gets detected, and digitized. So you have a powerful transmitter and a very sensitive detector or receiver to do this. And they're not operating at the same time. Either we transmit or receive. Sometimes we're also doing NMR on the same time. So we're not just hitting the electron spins, but we're also hitting the magnetic nuclei. For example, we could do proton NMR at the same time. So we have a built-in NMR transmitter in the Pulse EPR spectrometers. And this method where we do both at the same time is called ENDOR, for electron nuclear double resonance. It's kind of a combination of NMR and EPR. So that's how Pulse EPR works. Now here's the most basic experiment. It's called a free induction decay. Look at this diagram here. So first we're gonna apply a pulse. We're starting out at thermal equilibrium. All the spins, all the magnetic moments align with the field, just like compass needles in their lowest energy state. This blue arrow indicates a group of spins, like from all our proteins in the sample. Now, if we apply this 90-degree pulse, we tilt the spins so that they all point sideways. Okay, and this is over within a few nanoseconds. Now, since we're now sideways, we're in a non-stationary state, and those electrons will start to precess like this. Now recall that we're in a solid-state sample. Everything's inhomogeneous. The resonance frequency of every protein is slightly different due to orientation and unresolved splittings and so on. Which means over time, the different spins we flipped or have mutated will precess with different frequencies, and they will go out of step. Okay? One, there will be spins which have a high resonance frequency and the spins with a lower resonance frequency. They get out of step. And the response we get, we measure with the detector, is the vector sum of all these spin vectors here in this plane perpendicular to the static magnetic field. So if you see here, they point all in the same direction. They all add up. We get a strong signal. And that's what's called the FID. But as time passes, they start to fan out, lose what's called their phase coherence. And if you add those vectors up, you get less than if you add all those vectors up here. So it decays. That's why it's called a free induction decay. It's free because we're not doing anything to it. It just happens without our doing. An induction decay. Decay is obviously induction is an electrical term. Okay, this is called an FID. It's the most basic NMR experiment and works exactly the same in EPR. Except NMR works on a microsecond to millisecond time scale, and EPR works on a nanosecond time scale. So it's all like super fast. Here is the next step up of a Pulse EPR spectrum. It's called the electron spin echo. This Pulse sequence has multiple names, primary echo, Hahn echo, or so on. So imagine we got this FID. Now these spins are still precessing, right, in our ensemble of protein molecules in the sample. So they're completely dephased. But now at some point we can apply a 180-degree pulse. We basically take each spin and rotate it by 180 degrees. And now if you imagine the red spin having been the ones that was fastest precessing and the blue spin was the lowest, let's say, right, like this, red and blue. Now we're going to swap them around. The fastest spin gets a penalty, and the slowest spins get in front. So now if you let them again precess after the pulse, the fastest spin will catch up with the slowest spin, and at some point, exactly after the same time tau, there was the space in between these two pulses, at this tau time after the second pulse, all the spins will come together, they will rephase and point in the same direction. And that will give a signal, okay? It's kind of like undoing the FID or redoing the FID backwards, right? It goes up. This is a reverse FID. Then once we're at the top, it's again an FID. And this is called an echo. It's like two FIDs back to back. So we're doing spin gymnastic. And what's the purpose of this? We're going to hit it at a certain field, certain frequency, and see how large our echo is. So in Pulse EPR, all we do, we measure the amplitude of this echo. If the sample is diamagnetic, there is no echo, right? If the sample has a lot of spins in it, the echo will be large. And now Pulse EPR essentially measures things as a function of field. All right, so this is kind of the very basic Pulse EPR experiment. And you know, the other, like a few years ago, I was in Rome at the Vatican, and I saw this picture, right? This was painted by Raphael. And you know, historians say this is like the school of Athens, right? All the philosophers are painted here, and this is Plato and Aristotle and a bunch of others, Diogenes and so on. But I don't think their assignment is correct. I think this was the 0th bioinorganic workshop. And the reason is this. Look at Plato and Aristotle. Plato points up, right? Initially, the spins are in thermal equilibrium. And Aristotle says, yes, after 90-degree pulse, they go into the xy plane and they start dephasing. Okay, yes. But then there was also Michelangelo who was present as a student. But, you know, he just said, oh, darn, I forgot to have coffee. Then the older gentleman here on the right is Socrates. And he was Greek, right? And he was a little out of his home waters. And he looked at this menu, beer menu, and it read double IPA. And he had no clue what it means. Does it mean having like 2 pints of IPA? Okay, but the final conclusive evidence that this was actually a bioinorganic workshop is looking at this gentleman on the back here. There. Looks a little not like he looks today. That is, come on, Karsten. And you know why I know it is? Because you can read his mind. Look at him here. And you can very clearly see what he's thinking. All right, so that's the 2-spin echo, seen back in the day. All right, so now let's move to the second topic here, which is actually now we can look at these things by doing the spin gymnastics. And that's where we have to look first at magnetic nuclei and their interactions. So there are actually 3 important interactions we have to be aware of when we look at magnetic nuclei. They affect the energy of the magnetic nuclei. The first one is the nuclear-Zeeman interaction. That's the basis of NMR. It's the magnetic interaction with the externally applied magnetic field. The second interaction is extremely important. That's the hyperfine interaction. That's a magnetic interaction between a magnetic nucleus and an electron spin that's nearby. And there are multiple contributions to this hyperfine interaction. It breaks down into a through-bond contribution, which is isotropic. It's called the Fermi contact hyperfine coupling, Fermi contact interaction. The second contribution is through space. So it doesn't matter whether there's a covalent bond or not. It's the same. It goes straight through space. That is anisotropic. It's a dipole-dipole coupling. There's a third component that's due to spin-orbit coupling, but it's not relevant for many Pulse EPR experiments. Then the third interaction is actually an electric interaction. There's interaction between a non-spherical nucleus and an inhomogeneous electric field. This is really valuable in certain cases, but much, much less important than a hyperfine interaction, which carries most of the information you want to get from Pulse EPR. So people often call Pulse EPR for bioinorganic purposes hyperfine spectroscopy. It's a way to figure out hyperfine coupling. All right, so let's briefly talk through number 1 here. The resonance frequency of a proton is proportional to the magnetic field and to the nuclear G factor. So for example, if you have a proton at a magnetic field of 1/3 of a Tesla, and that's what we're going to use in a practical, that's a typical field for EPR, the resonance frequency is 15 megahertz. Now, if you've done NMR before, you've probably run on a 400 megahertz or 500 megahertz machine. And the fields are much stronger. Now you would say, oh, why these EPR people use this really weak field? WIMPs, right? But it turns out we can't go much higher because the electron resonates at extremely high frequencies. The magnetic moment of the electron is about 660 times stronger than that of a proton. That means we have to lower the field, we have to up the frequency to get it into resonance. Now, different nuclei have different GN factors, so they resonate at different frequencies. They also have different spin quantum number, as we've seen yesterday. We looked at copper, had a spin 3/2, or manganese had a spin 5/2. Iron 57 has a spin 1/2. So these are metal nuclei that are magnetic. But they're also the most common ligand nuclei, hydrogen, nitrogen, carbon, oxygen, phosphorus. And you could also add sulfur that have spin and have GN factors. And Pulse EPR mostly goes after these protons and nitrogen and oxygen and phosphorus nuclei. So hyperfine coupling. That was the second of the third relevant interactions. And that broke down into multi-components, and one is the Fermi contact interaction. If you look at this equation, you can disregard those prefactors. The important thing are the colored quantities. One, the red one is the spin density at the position of a nucleus. And the blue one is the GN factor of the nucleus. The red quantity is the one that changes from molecule to molecule. And as you know, electrons are delocalized in orbitals. And some of these orbitals can delocalize and put some non-zero spin density at the position of a ligand nucleus. Right? And that's what we can pick up with this Fermi contact interaction. You can have there's a reasons for getting this there. You might have a ground state, open S shell state, or way more often you have a valence core spin polarization. So let's say there's an alpha spin floating around in a valence orbital, and then that polarizes the core orbital to put a beta spin on the nucleus, right? So here's a very simple interpretation. Look at the first line in this table here. Take a proton with a spin 1/2. If it were just a proton with a 1s orbital with an unpaired electron in it, just a hydrogen atom, simplest possible thing, you would measure a Fermi contact hyperfine coupling of 1,420 megahertz. Okay, so that's our reference. That means like 100% of the unpaired electron is in that 1s orbital. Now, in your protein you're studying, maybe that's not the case. Maybe you measure a coupling of 20 megahertz. Now you can just do a very simple, back of the envelope calculation. 20 divided by 1420 is about 1.4%. So you can say that proton sees the electron spin for 1.4% of the time, or then a 1.4% spin population is out there. So what can you learn from these isotropic hyperfine coupling? You can learn about spin delocalization, whether your unpaired electron is centered at the metal or it's like washed out onto the ligands. And you can also test the proximity of the nucleus relative to the metal ion, knowing if you know your structure. So that's the first component. The second component is the through-space dipolar coupling. And that's independent of any orbitals delocalizing over to your nucleus. Here's the electron spin, and then through space, this, picture a big protein here. There's your magnetic nucleus, and each magnet, both the electron and the nucleus, are associated with a dipole magnetic field. You can draw those field lines, and what you realize then here, if you're at the position of the nucleus, you will see that it feels a field that's due to the electron. It points this way. We call it the hyperfine field. Okay? Now, this hyperfine field can be very strong. It can be also very weak, and it depends on two things. It depends on 1 over r cubed, where r is the distance. So if I move this nucleus twice, double the distance, this interaction strength will drop by a factor of 8. So we kind of have a lens on our system that drops cubically as we go away from the electron spin. It also depends on that angle theta, which is the angle between the external magnetic field and this interspin vector, as we call it. And if this magnetic nucleus were here on top of the electron, kind of parallel to the field, the interaction would be twice as large if this, compared to the case where this proton or nucleus would be in a way perpendicular like this. Okay, now, of course, the nucleus will feel two things, right? If you put yourself in the shoes of the nucleus, you will feel the hyperfine field from the electron spin and the external Zeeman field, static field. So you see a total magnetic field. And that's the one we're basically, that you're going to be equilibrated with. That's the field that's relevant to determine the NMR frequency. Now what happens if you flip the electron spin, right? We're going to flip the spin with a 180-degree pulse down. The field lines, instead of going this way, like up, out, and down in, it will go the other way. Then the hyperfine field will also flip and go in the other direction. So now if you look here, what happens is we flip the spin, the hyperfine field is flipped, and the total field points in a completely different direction. Right? And so this is kind of the interactions between electrons and nuclei that we're probing with Pulse EPR. This is the very important thing. Now, how do these spectra look like? We're measuring now NMR spectra. We give them other names, but basically these are just nuclear spectra. We see as a function of a frequency, which we apply to the protons, for example, we see peaks. These are really true simple absorption spectra. Here is the very simplest case of a single protein. You will see, here is the, let's say this is a proton frequency, 15 megahertz, remember 2 slides ago. Now, if this proton is coupled, is nearby an electron spin, it will have, you get 2 peaks. One from those molecules where the electron spin is up. That will be a frequency up here. And another peak from all those protein molecules where the electron spin is down. Right? So that will be this peak. So this is a Pulse EPR spectrum, is basically the sum of 2 NMR spectra. Proton NMR with electron spin up, and proton NMR with electron spin down. And this is just for a single crystal, right? But typically we have these powders of frozen solutions, and since this coupling is orientation dependent, we will get everything at the same time. We get a really broad NMR spectrum, solid state NMR spectrum, okay? And that's actually really great because we can now use these shapes to tease out what's the contact interaction, the through bond interaction, and what's the through space interaction. And both hold separate valuable information. Okay, then there's the third interaction I told you about, nuclear quadrupole. That's of minor importance and of generally minor importance, but just briefly summarize what it's due to. Nuclei with spin greater than 1/2 are non-spherical. They have a non-spherical charge distribution. And the spin is tied to that shape. So for example, you take a deuterium nucleus, it's kind of egg-shaped. If you take a carbon oxygen 17 nucleus, it's burger-shaped. It's flatted out. Okay? These are called quadrupolar nuclei. They're non-spherical. The consequence of this is now if you put this non-spherical charge distribution in a molecule, there will be electrons and other nuclei around, so we have an inhomogeneous electric field, which means that this non-spherical nucleus will feel a torque to align it with like the electric field gradient locally. So for example, this here will be the highest energy if these are positive charges and these are negative charges. This is a high energy state, right? If you turn it a little bit, it will torque this way until the positive charges and negative charges are closest. This will be the lowest energy state. Why do we have to worry about this? Well, it's an electrical interaction. It seems like we're doing magnetic resonance. Why do we need it? Well, the spin is tied to the nuclear shape. So if the nucleus turns, the spin will turn as well. Okay? So that's the third interaction. So now what are the types of pulse experiments? Generally, they work this way. You generate a spin echo, or an FID, and you measure its amplitude as a function of something. Okay? And depending on that something, you get different types of spectra. You could just do a field sweep, just like for CW-EPR. Sweep the field and plot on the vertical axis the echo amplitude. Then you'll get a so-called echo-detected field sweep, and that's just an EPR spectrum. It's the Pulse EPR's analog to CW-EPR. And that's the first experiment you always do, to check whether there are spins. Now, you could fiddle with the pulse delays. For example, you could change that tau value between the first and the second pulse. As a consequence, the echo will move in time, right? But it will also change its amplitude. And there is an effect called ECEM, electron spin echo envelope modulation, where this echo amplitude is modulated by the nuclear frequencies. So this is one way to measure nuclear spectrum, which is the data we want to go for as bioinorganic chemists. And there's a whole zoo of acronyms. There's a 2D experiment called HiScore. Okay, there's a second class of experiments that give nuclear spectra called ENDOR, electron nuclear double resonance. That's where we use the NMR spectrometer at the same time. We're going to flip the electron spins, and we also send pulses to the protons. And we get the type of spectra I've shown you before. Where are we here? We get these type of spectra. Again, these are nuclear spectra. That's how we change the frequency of the nuclear NMR pulses. We could also try to hit different transitions for the electrons using 2 different microwave frequencies. And that's then called ELDOR, electron double resonance. And that can give, it's an ELDOR-detected NMR that can also give you a nuclear spectrum. There's also a version called DEAR, double electron-electron resonance, uses 2 microwave frequencies. You can measure the distance between electron spins. So this is kind of the alphabet soup, but the takeaway here is we're measuring the spin echo as a function of something, and typically we get nuclear spectra, right? And then we analyze those peak positions in the nuclear spectra to learn about hyperfine coupling and learn about structure. So here are a few examples. This is an old example from 1976, Mims and Peisock. They looked at copper protein stellocyanine, and very little was known about these blue copper proteins back in the day. What they did, they measured the spin echo, vertical axis, spin echo amplitude, as a function of tau, that spacing. And what they saw is that they saw modulations. This is this electron spin echo envelope modulation effect due to nitrogens. So they were actually measuring the copper electron spins and saw modulations. Now they made some control samples. They made copper complexes of hexaglycine. They made a glycyl-glycine copper complex. They took BSA and added copper, and they made a copper imidazole complex. It turned out that this modulation looks more like the copper imidazole complex modulation or BSA modulation, and not like a glycyl-glycine coordination. So they concluded that there's an imidazole ligand to copper, and not an amides nitrogen from the backbone, which was at the time novel information, right? So if you have no structural idea, take an EPR spectrum and you learn about what is coordinated to your paramagnetic center. So the technique here was nitrogen-14 ECM. Here's an example from the 80s, also a very prominent milestone in Pulse EPR applications to bioinorganic chemistry. It's about a conitase which catalyzes an isomerization reaction. And here you can see the EPR spectrum. You can see the very typical rhombic spectrum we've looked at yesterday with the 3 feature, like the positive slope feature, the negative slope feature, and then the maximum feature. So if you now do ENDOR on this, measure the nuclear spectrum with an O17, you prepare this sample in O17 water. Expensive. But unfortunately, O16 is just non-magnetic, so you can't pick up an NMR spectrum. You have to isotope label. That's kind of the secret. Really always think about, okay, if I want to learn something via NMR or EPR about ligands, I have to think about magnetic isotopes, right? So here in this case, they added water with O17. O17 is a spin 5/2, and they measured in the ENDOR spectrum, and they saw a very strong hyperfine coupling. And strong means interaction is they're very close. And they saw like a multiplet structure that corresponds to spin 5/2. And that made them assign this to oxygen 17. But now, how can the hyperfine coupling be large? Well, where's the unpaired electron? It's on this cubane iron-sulfur cluster, right? So this water must be binding to the iron-sulfur cluster. They did the same experiment with substrate, citrate, and they could basically conclude, without a crystal structure, that both a water molecule, at least one water molecule, and citrate are binding to the active set, which is the iron-sulfur cluster. So again, structural information, coordination information from a nuclear spectrum acquired with Pulse EPR. Here is an example from the late 80s about cytochrome C peroxidase. That is a heme protein with a histidine ligand, and there was this state called compound ES that was very poorly understood. It was thought to be a state where the heme paramagnetic center is interacting with some close nearby organic radical. And it wasn't clear what type of radical this was. So what happened, this is work from Brian Hoffman. What he did, he then prepared, he just measured proton and deuterium ENDOR on this sample to figure out and see how the spectra look like. Well, here on top you see the proton spectrum. Here it's centered. You see it's sort of symmetric. And remember, typically you get like 2 peaks, right? And it's sort of symmetric. There are some intensity distortions that are of instrumental nature. But then what he did, he mutated. He thought this might be a tryptophan. So there were 2 tryptophans that were close to that heme center, a 51 and 191. Mutated the 51 to a phenylalanine. The spectrum did not change very substantially. All the features were still there. that this is the proton. See this arrow here doesn't mean spin up, it just means look at this axis. Okay? Then he labeled the tryptophan 191. Actually, he labeled all the tryptophans with perduterated them in proteated solvent. And what he saw that some, like this feature went away, see it's gone. This broad peak here went away. So clearly we're picking up a loss. We're losing these hyperfine couplings by removing the protons from the tryptophans, right? Now this tryptophan had no effect, so it must be this, okay? And so to make sure, he also looked at the deuterium spectrum. Here you look at the bottom axis. This is deuterium NMR. And you see there are peaks. Without the deuterated, there are no peaks, and he could clearly assign the radical he saw to tryptophan 191 and basically figure out what the electronic structure of compound ES is. Here's another example from the early 2000s from an enzyme, pyruvate formate lyase activating enzyme. It's also an iron-sulfur cluster, and it uses S-adenosylmethionine as a cofactor. Here are the EPR spectra. I showed them yesterday as well. Without SAM, you see a certain spectrum. You add SAM, the spectrum changes. So clearly, SAM is coordinating to that iron-sulfur cluster. But how? Okay, that was the question. Crystals were not possible, so Endor was the solution. So what was done here, again, carbon-13 and deuterium Endor. And what you can see, maybe let's look at the carbon-13 first. Carbon-13 is a spin 1/2 nucleus, and so you would again see two peaks. And they measured the Endor spectrum as a function of field, and you see here is the center. The center is always the Larmor frequency, the NMR frequency of the nucleus you're looking at. And the splitting, that's the reports on hyperfine coupling. Now that's indicated by red. That is the Fermi contact splitting, Fermi contact interaction. Tells us how much spin density is on that carbon. And it's actually substantial. It's noticeable at least, right? We can resolve it. And then these peaks have a width. And that width is due to the anisotropy of the hyperfine coupling. And that tells us about the through space coupling. Remember, hyperfine coupling, you have through bond and through space. So the red one gives us the through bond coupling strength, and the blue widths here give us the through space coupling strengths. And from these data, analyzing just this single spectrum, you can say that the carbon-13 we're looking at, and it was a carbon-13 labeled S-adenosylmethionine, it must be 4 to 5 angstroms away from that spin center, which was the iron-sulfur cluster. You could also back this up with deuterating the protons on a metal group and look at the deuterium Endor. So again, here we not just identified that the ligand was coordinated, but also how. Because we also got the distance between that center and the protons on the methyl group. So you can kind of do ligand crystallography with it. Marty?
Yeah, I'm just going to ask, in that particular paper, I don't recall how they decided what the distance was from. I know what it's to from the measurement, but is it to the center of spin in the iron-sulfur cluster, or is it, and was that decided by DFT, or?
So it wasn't, it's still open. So 4 sulfur clusters are computationally very challenging. And so what you typically would do if you have a delocalized electron spin to calculate the through space interaction, you would have to, let's say for simplicity, let's say there is 50% spin here, 50% spin here. You would just have to add up 2 contributions, 50% dipolar coupling from this iron to the hydrogen, and another 50% through this, right? So what's called a distributed dipole model. So sometimes people analyze it that way. I don't think in this particular paper he had a particular clue which ones of these were. But the distances were so short that the conclusion that it's directly coordinating was fully supported. Okay, but iron-sulfur clusters, breaking them down is impossible. EPR on iron-sulfur clusters looks like this. It's a fingerprinting exercise. We don't know much. We can't quantitatively calculate the G-values. Hopefully in the future at some point. Okay, here is another story about, this is actually from very recently. It's the same enzyme. Here's a 2016 paper on pyruvate formate lyase activating enzyme. And here they were able to trap an intermediate state. And that intermediate state is where this S-adenosylmethionine is cleaved, reductively cleaved, and gives the 5' deoxyadenosyl radical. And using carbon-13 Endor, and you can see here's again the nuclear spectrum, nuclear frequency, right? You can see the center, that's the Larmor frequency of the carbon-13. You see a peak on the left and a peak on the right. And by looking at the splitting, you get the isotropic coupling, the Fermi contact. And by looking at the width and the structure of the peak, you can get the through-space coupling. Right? And from these is, again, carbon. They could figure out that this carbon-13 from the, where is it here? The carbon-13 from this methionine, from the adenosine, must be directly coordinated to the iron-sulfur cluster. And that's their kind of artistic rendition of this, or how this should look like. Okay, so directly identifying a carbon-iron bond, if you will. Here is an example from a nitrogenase, and Oliver Ainsley has done this work together with Stefan Weber in Freiburg. And here, this is the FeMoCo, the iron molybdenum cofactor nitrogenase. And there, at the center, there's this light atom, carbon, nitrogen, oxygen. It was long unclear what actual identity was. And one of the pieces of evidence that showed that this was a carbon came from ECM. Look at the center trace and look at the black trace here. This is, again, a nuclear spectrum. It's recorded via ECM, not via Endor. That just gives different intensities, but it has the same peak positions. You see, the wild type didn't have any feature near the carbon-13 region. Right? But then if you grow this enzyme carbon-13, you get a large peak at the carbon-13 Larmor frequency. That's probably due to all these other ligands here that see some of the electron spin. But then you see this little bump here and this here. A peak to the right and a peak to the left of the Larmor frequency. 2 peaks again. And the splitting is really large. And that was supporting evidence that there's like a carbon really in the midst of that spin density on this FeMoCo factor. And it's a very clear demonstration that what was going on. They also did an N15 experiment to show it's not a nitrogen. That's an important control experiment. And they didn't see any new features coming from nitrogen. So you got a carbon-13 signal, but they didn't see an N15 signal. So it was a carbon. Of course, together with Oliver's analysis of the electron density and Serena de Boer's X-ray spectroscopy that nailed that down. Another application is actually, if you look at photosystem 2, which contains this manganese 4 cluster, of which we now have a crystal structure. About 15 years ago, nothing was known about this. We didn't have a crystal structure. Here are very complicated EPR spectra. This is work from Dave Britt, and he did manganese 55 Endor. Manganese 55 is a spin 5 half nucleus, so it gets really complicated. And since the electrons directly sits on those manganese nuclei, the coupling is really strong, and the frequencies are hundreds of megahertz. And analyzing the shapes and positions of those peaks, Britt was able to actually come up with a model, a spectroscopically derived model, that there must be 3 manganeses that are like talking to each other more strongly, and the 4th one should be a little further away. And he called it a dangler manganese. Okay? And surprisingly, the crystal structure bore this out 10, 14 years later. So that's another way of doing it. If you have a metal cluster, sometimes Endor or ECM can help you figure out something about the structure of that cluster. Here is work done by Alexei Silakov, who is running some of the EPR tutorials here. It's on hydrogenase, iron-iron hydrogenase. And there is a dithiolate ligand connecting these two, I'm showing here a model complex, but it has a di-iron center in it with a dithiolate ligand. And this atom there on the top, which now is known to be nitrogen, was completely unknown. Is it a carbon? Is it a nitrogen? Is it an oxygen? And what Alexei did, he did high-score, which is a 2-dimensional Pulse EPR method, very akin to 2-dimensional NMR, and he looked at nitrogen-14 and nitrogen-15 signals. And by analyzing the isotope substitution pattern, pulling out the hyperfine couplings, and also doing some deuterium and proton and Endor, he could clearly show that the bridging atom there is a nitrogen. So now we know it's an acid dithiolate, as opposed to have an oxygen in there or sulfur or something. So again, this helped Pulse EPR here, via looking at nuclear spectra, helped us identify the nature of the ligand. So here is something that did not have Pulse EPR. And I bring that up because it's answering the same questions. What ligand are we looking at? What spin center are we looking at? This is from an enzyme called lysine 2,3-amino dismutase, which converts alpha lysine to beta lysine. And this is work from the very early 2000s of Perry Fry. And what he did, well, there's this postulate that there's this 5' adenosine radical intermediate. And it was just so elusive. Nobody could catch it. What he did, he made a cofactor analog with a carbon-carbon double bond, so that once you get to that state in a reaction, that radical, the unpaired electron would be delocalized over a few carbons and become a little more stable. And this way, basically got an intermediate analog that could be studied with EPR. So then here you see just continuous wave EPR spectrum, but the same idea. Analyze the spectrum and see what happens if you switch out isotopes. Okay, when he then took these cofactor analog and did site-selective, position-selective deuteration, he saw this spectrum change all the time. By analyzing and simulating this spectrum, you see that in dashed lines, you could clearly assign this to a allylic radical of an anhydroadenosine radical, basically. And that was kind of the first evidence that there is some sort of a radical intermediate in these reactions. This is CW-EPR, but see, it's all about hyperfine couplings. And you can nail down hyperfine couplings and identify them and assign them with isotope substitutions. Okay, here is work, again, here from Penn State, great work on CFR, where people just looked, I think this, again, Alexei has done a lot of this work. Use both CW-EPR and Pulse EPR. Here you see Endor spectra, proton Endor spectra. These are deuterium Endor spectra, and again, proton Endor spectra. Analyzing the couplings, the splittings, the width of the lines, you could identify the nature of the paramagnetic substrate, the substrate radical, and very clearly assign where the unpaired electron is. To wrap up, I'm going to show you two more examples, but this time for a technique called DEAR, double electron-electron resonance. That is kind of the electron-electron analog of the electron-nuclear coupling, which we call hyperfine. Now, there is no special name for electron-electron coupling. We just call it dipolar coupling. Here's a very complicated metalloprotein. That's from complex 1. And you see it contains, 8 iron-sulfur clusters and a flavin. And depending on what kind of conditions you produce this and you put this into, even electrochemically, you get a ton of different EPR signals, and some of them are shown here. And it was completely unclear which EPR signal comes from which iron-sulfur center. And that's where pulse EPR spectroscopy could provide an answer. So this is work by Judy Hurst. And what she did, she poised the system into specific redox potentials to get a mixture of EPR spectra, and then used the pulse EPR method to measure the distance of those mixtures, of those, let's say, I'm not exactly familiar with all the details, but some of these spectra have like 2 main components. Right? So there is clearly 2 radicals at the same time in a protein. By measuring the dipole-dipole coupling, she could pull out the through-space interaction strength and this way the distance. And using the distance and the crystal structure, which is known, she could assign the EPR signals to the specific positions of the iron-sulfur clusters. And here's the table. 8 clusters, I think 4 or 5 EPR signals, and the assignments by Judy Hurst, based with using double electron-electron resonance. The last example I'm going to show is actual work that I did, where we look at cytochrome P450 CAM, the bacterial cytochrome P450, and here I used nitroxide distance measurements to measure not the active site geometry, but the overall conformation of the protein. So if you're more into structural biology, pulse EPR is also useful for figuring protein structures out. So what we did, the question here was, is a globular protein, there's an active site at the center, and there's a kind of a clamshell motion, right? The question is this clamshell open or closed without the substrate? Or is it open or closed with the substrate? It was very simple to do. We site-specifically put nitroxide radicals on 2 sites on the protein, on the 2 parts of the clamshell, and then we measured the distance before the addition of substrate and the distance after the addition of substrate. These are the raw data, and here you see what typical DEAR data look like. There's a horizontal axis with the distance, a vertical axis is kind of a histogram. And you see there's a dominant distance of 5.6 nanometers without the substrate, and a dominant distance of 4.9 with the substrate. So we clearly see the protein is closing around the substrate when you give it to it. All right, so to summarize, what is Pulse EPR? Pulse EPR is a set of high-resolution EPR techniques to determine local structure around a spin center. Metal ions, metal clusters, or radicals. In contrast to CW EPR, which only works very close to that metal ion you're generally interested in, Pulse EPR gives you high resolutions. You can resolve weakly coupled magnetic nuclei. You can look at ligands in the first and the second ligand sphere. You can actually measure hydrogen bonds. You can measure distances to other spin centers. And it works for magnetic nuclei within a few angstroms of the electron spin, and for other electron spins that are within a few nanometers away. Okay, then here is a big summary of what you can learn from EPR data. If you do CW or Pulse EPR field spectrum, you can learn about the type of the spin center, the oxidation and spin state, coordination geometry, electronic ground state, and spin delocalization. If you do nuclear spectra, either ECM or ENDOR, you can learn about the type of the nucleus, the protonation states, the location of the ligands, the coordination mode of the ligands, and you also can do oxidation state assignments within a cluster. And lastly, if you do measure dipolar spectra, you can measure the distance between spin centers. So that's the bottom line of what Pulse EPR can do for you. And I hope that gave you a little bit of an overview. As I said, look to the 2014 slides if you want to learn more about the spin physics. Thank you very much.
Now Dennis is going to give a more specialized application of this, and you see the type of there. Thank you, Dennis.
Thank you, Karsten. Well, I'm glad that we made here too. It was a long last several days, more like 2 weeks. As of last night, we were in East Lansing, and now we're back here right about 3 o'clock in the morning last night. So the talk that Karsten asked me to give is on continuous flow spectroscopy, and that's what you find in your description. But I think I will de-emphasize that, and notice I put that in the parentheses here, more toward a time-resolved spectroscopy and specifically isotope difference Raman spectroscopy. Unlike some of the other presentations you've seen here, it will be more qualitative. My goal is not so much ask you what can you do for Raman spectroscopy, but rather what Raman spectroscopy can do for you. And we'll come back to that question at the end. Because of the very nice introduction lecture given by Frank Nies on the last workshop, I'm not going to go through details of that. I'll just do a quick refresher. And then we'll go for a number of examples, hopefully going a little bit low and slow so you're more comfortable with that. So first of all, definition of time-resolved Raman spectroscopy or any spectroscopy with Raman specifics. That's generic description of a reaction, right? So you have the enzyme, typically an enzyme, the substrate or cofactor. They will bind, proceed through a number of steps, eventually releasing the product. And generally, our goal is to understand what is that sequence of detectable steps. The steps that, of course, must populate in the process. So there are several factors that are common to other techniques and specific to the Raman. First of all, you have to trigger the reaction. And as with any other time-resolved approach, unlike, for example, EPR, when we're talking about time-resolved EPR, when you have electronic control, here you have to start with the reaction itself. So, and there are two general ways of doing that, either by photolysis, if your reaction photosensitive, and there are a few applications, lucky applications that are, or typically by mixing reactant and product. Once the reaction has started, you have to define the phase of the reaction you're looking at, and you have to stabilize it. You can do it, for example, for photochemical reactions by the probe delay. You can use quenching delay for RFQ or something like that, or chemical quenching, or you can do it by spatial resolution based on the continuous flow methods. After a certain delay time, you have to excite the molecule. That's very specific for the Raman. You have to bring the laser flash. There is no variability here. You have to have a laser, and the laser has come to the sample. Because of that, there are always a threat of reaction by products. So whenever you deal with a resonance Raman or Raman spectroscopy, even though we don't emphasize that in the literature, always the question in the back of our mind, are we photolyzing the sample? Is what we're looking at real, or is it not? So we always have to go extra half a mile at least to ensure that we're looking really at these intermediates and not at these byproducts. And it can get very complicated if that's the way it goes. Then you have to detect the species, as with any other technique. And in Raman, the difficulty is that the ratio, typical ratio of the number of excitation or relay photons versus the Raman scattering photons is about 10 to the 7th. So we're looking for very weak signals, very small signals, and that's for a single frequency. Of course, we want to spread it in a spectrum. So we have another order 2, 3 of a loss if you're looking at the broad spectrum. And there are different ways to overcome that, and one of them is to do rapid freeze quench, where the reaction is stopped. So you think you have a luxury of measuring that indefinitely. Typically, you don't. Or you can do it by the continuous flow, where the sample is continuously delivered to the sample point, refreshing what you're actually measuring. And lastly, you want to identify what you're measuring. To detect the Raman signal is the first step. It's maybe a quarter of the story. The other 3/4 is to make sense out of what you're seeing. And we're using isotopes, we're using kinetic information and different models to accomplish that. So because of this factor, non-Him Raman spectroscopy is typically a noisy spectroscopy. And I should warn you that you're going to see a lot of noisy spectra today. And not that they are low quality, that's a very common. It's not like a porphyrin where you can measure Raman by almost eyeballing the sample. Principle of Raman spectroscopy is if you have electronic transitions, ground in the first excited electronic state, you can bring the photon will interact with the molecule, reach a virtual state, and then release a photon. Most of those photons will come right back with exactly the same energy. Those are relay photons. But a small fraction, 10 to the minus 7, will leave a little bit of energy at the molecule. And we're really interested in those small differences. You can see that this is complementary to the IR absorption spectroscopy, which is also looking at the vibrations. And we're trying to interpret that structure. So if the energy of the photon matches energy of electronic transition, we now can start talking about resonance Raman, which increases the absorption efficiency and increases the cross-section of Raman signal, helping great deal. And an example here is a generic porphyrin spectrum. There are two important dependencies here. Is first of all that the intensity of Raman signal is proportional to the 4th power of the wavelength. So the farther in the blue you are, the better. And second is that it is scaling approximately with the square of the extinction coefficient. Now you can see why it is such a big deal between porphyrin system and non-porphyrin systems. So this is a general process. There are a few advantages and disadvantages. Raman can clearly do a low frequency measurements, which is very difficult in the IR. It's not sensitive to water or optics. We can measure that in aqueous solution, no problem. We can use both the pulsed and mixed time-resolved approaches. And Raman is selective. It can be a blessing, it can be a curse. If you're looking at the infrared spectra of complicated protein, it may be a mess to make a sense out of it. And certainly water and optics are much more complicated, and particularly for the metal ligand vibrations, which typically appear in the low frequency region, not accessible to the IR, except for the very specific types of IR. There are a few other things that may be going on here. And for example, we can have relaxation of the excited state, and we can generate a fluorescence. Notice it is in red. Fluorescence is not a good friend of Raman spectra, right? So it can overwhelm what you're measuring. You can also have a chemistry going on, as I mentioned on the previous slide. So we're always aware of those things and try to solve that. That can be solved perhaps biochemically, by changing the isolation, and these can be solved by spectroscopic means in part. So how do we deal with the vibrations? The simplest case is a diatomic oscillator. And here we're looking at an example on iron and oxygen, for example, visualized as two masses connected by a spring. And they're vibrating, of course, as you know. The frequency of this vibration is described by this relationship where K is the force constant of the bond that you're looking at, and M is the reduced mass. In the case of diatomic oscillator, this is the expression for the reduced mass. So looking at that, you can immediately predict if you're going from oxygen 16 to oxygen 18, that the frequency A will decrease. If you go to the heavy atom, the frequency will decrease, proton, iron, and so on and so forth. And it will decrease by a predictable manner. So both the frequencies and the amplitudes of isotopic shifts are meaningful for what we're doing. That's just about all you need to know. In reality, we rarely look at the absolute spectra. So usually we'll deal with the isotopes to identify what is what. Here we have a generic diagram of a spectrum that contains both isotope insensitive band and the isotope sensitive band. Raman spectra can be very rich. We don't want to assign all the bands that are not relevant to us, so we simplify it. We take the difference between the two spectra, and of course these bands will cancel out, giving us a nice derivative pattern. So what we want to know the frequency, typically by the 16 oxygen isotope, and the magnitude of the 18 isotope downshift, which helps us with assigning whatever particular modes we have. So here is a somewhat of old example, case of cytochrome C oxidase. It's a membrane protein. It contains two porphyrins, and here is the good illustration of power of Raman. You can selectively excite one or another. So you can see, even though both of them are very similar cofactors, you can selectively observe structural changes at one or another by tuning the wavelength across the solar region according with the individual absorption. Of course, if your cofactors have exactly the same absorption, it doesn't help too much. So in this particular case, we're very fortunate. We can start with the enzyme which is poisoned by carbon monoxide. And in the dark, carbon monoxide dissociates in about 30 seconds. So we can mix the poisoned enzyme with O2. Nothing will happen for about 30 seconds. We have something useful to do in the time frame. And the useful thing is we can dab the protein with a red light, which causes dissociation of CO. And now on the way back, oxygen outcompetes CO by several orders of magnitude. So it will generate an intermediate species, and then we were waiting a certain period of time between initial flash and the probe flash. This is pump and this is probe. And we observe Raman spectra. As I showed you here, those are isotope difference spectra, so we'll be looking at the patterns that will look something like this. All of those noisy squiggles, actually, this is a very high signal-to-noise ratio Raman spectrum, if you're wondering. So all of these squiggles are isotope oxygen vibrations involving oxygen one way or another, which is great. We have frequencies, but it doesn't really tell us what the structures are, right? We can guess, but for example, in this region, we have multiple different structures that will give approximately that frequency and approximately that isotopic shift. We cannot distinguish them by the frequency or the isotopic shift itself. So what do? Again, we use isotopes. And here is an example. Specifically for the 800 wave number region, there are two plausible structures for the porphyrin. One is a terminal iron 4 oxa, and another one is iron 3 pyroxa. And there's been a debate in the literature for a very long time whether one or another is populated in cytochrome C oxidase. But notice what happens. This structure contains a single oxygen atom. If you start with either 16 or 18, of course, it will be that isotopomer. However, if you start with a mixed oxygen isotope and one of the atoms is heavy, you have a 50-50 probability of finding one or another. But once you do, the frequencies of those vibrations are exactly the same as for the symmetrical isotopes. So these frequencies coincide. In the case of OO stretch, that frequency depends on the masses of two atoms. So if you replace one of them, the frequency goes down by half the magnitude. So in this case, when we take a difference, a simple difference between 16 and 18 will look exactly the same. It's not going to help. But the double difference between symmetrical isotopes and the asymmetrical isotope, in this case, will cancel exactly, giving you a flat line. And in this case, it will give you a pattern that is indicative of new frequency appearing with a mixed oxygen isotope. So here's an example of how that was done. Notice that here that was done with the excitation at 607 wave numbers. I said it's not very favorable for the Raman, low intensity, a lot of fluorescence, but nonetheless we will like it to pick up those signals, and you can clearly see that with the 16-18 oxygen isotope we have exactly the same frequencies at the same positions. When you do the double difference subtraction, you get nothing. That nothing is a good indication in this case. It says unambiguously that the OO bond is cleaved. So this intermediate is a oxa, not a pyroxa. This intermediate, on the other hand, is the iron O stretching of a oxy species. And it does depend on the frequencies of both oxygen atoms, but it primarily depends on a proximal oxygen atom. So you don't get a single frequency as you have in this case, but you have a double shift depending on which orientation of your oxygen is bound at. So sometimes that is useful as well. Okay, so from the perspective of Raman spectroscopy of inorganic compounds and biological compounds, porphyrins are the best studied. We have a nice probe. The geometry is fixed. It has been very well characterized computationally and experimentally. So there is not much happening in the porphyrin fundamental chemistry here. Non-heme systems are very different. They're flexible, they're variable, and we have to do a lot more work to understand what is going on there. And there are two examples we'll take a look today. It's mononuclear non-heme iron systems and dinuclear non-heme iron systems. In both cases, we want to look at the isotopic substitutions as a way to make an assignment. Kinetic information, we can't do it based on Raman alone. Catalytic information, and what model compounds are telling us. So let's start with the mononuclear system here. And the system in question is tau D. Right? So it has a facial triad. There is a rough structure of the active site. Because it requires a cofactor, there is AKG bound to the iron, carboxylate, and 2-metazoles, and this is our substrate. Based on other techniques, we know there are several species en route from reaction start to the completion, and one of them certainly attracted our attention after the publication, actually, from Penn State, which is a putative iron 4 at that time, iron 4 oxo. So what do we need to address it? It's a good candidate for us, but before we can do Raman on it, we need to know A, absorption of that species, and B, time domain. Right? So if you call me and say, look, I would like to do Raman on my compound, these will be the two questions I'm going to ask you first. The third question will be how much protein can you produce? But that's not at one. Again, it was published by Penn State group that there is a transient increase in absorption in the neo-UV region. So for us, it's great. We have an answer to that question. We have the region where the intermediate absorbs. We can excite it. So second question is time domain. When do we look? So in this case, the reaction is substrate deuteration sensitive. With the detouring, it lasts a lot longer. It makes a natural target for us to go after. So in the combination of both the kinetics and the spectra, we have a nice region where to look, two-dimensional region, spectral region and temporal region where to look. So we want to look in somewhat in this region because we can look at both the detouring and the effect of deuteration protonation here, and we don't use as much protein. So we're using TR3 stands for time-resolved resonant Raman spectroscopy. In the simplest case, we can start with our protein. Oxygen, remember, it is a weak spectroscopy. We have to accumulate for a long, long time. So we continuously are flowing the sample through our Raman spectrometer, excited with the laser beam, and collect Raman scattering at 90-degree geometry. Then we can do the same with the 18-oxygen isotope, replace the sample, measure it again. And the problem is that we can measure that forever, but our collaborators cannot provide enough protein to do it in a real time because the time span of this reaction is on a millisecond scale. We can flow a lot of protein in a hurry, right, and then wait for years for more protein. So we decided to use Arrhenius relationship and slow down the reaction by lowering the temperature. And we've done it in this particular case. The temperature is minus 40 degrees centigrade. What is important to recognize, it is not a polycrystalline sample. It is a liquid sample. We're using cryoprotectants, ethylene glycol. Now, if you know what the consistency of ethylene glycol at minus 40 is, it's like honey. Right? So we are mixing two honey streams, and we want to look at the sub-second delay time. So it makes it an interesting challenge. So first advantage of that is that we can slow down based on the general activation energies by roughly 3 orders of magnitude, slow down the reaction, meaning we're reducing protein consumption by that much. But second problem is the viscosity, and to overcome that, we use an active mixing because the diffusion is not sufficient to initiate the reaction. It's not enough for us to mix the samples. So we use active mixing, which looks probably in a nutshell as your home mixer. Right? So if you mix flour with water, you are not really making a solution, but you make a homogeneous enough system where the diffusion will do the rest. And that's what's happening here, delivered to the flow cell. In reality, implementation is a little bit more complicated. So that's an AutoCAD design of the actual mixer that we're using in the lab. Sample point is somewhere here. Everything else is service to maintain the temperature, pressure, and so on and so forth, and achieve homogeneous mixing. That's how the earlier version of that looked like. So this is the same device. It is hidden, small piece hidden back there. The rest of that is sample delivery. Now it became a lot cleaner. We actually brought a new cryogenic cage. It is in the Raman room. If you're interested to stop by, take a look at that, how it is currently done. We used to do that manually. We realized we can no longer do that. The system became too complicated, so we're doing that by the computer. The overall system became much slicker now and much more reliable, which is probably more important. This is the close look at our system. This is our mixer. The beam is brought from the bottom. This is the collection lens. We bring the sample with the counter flow of cold nitrogen, pre-cooling the sample before the mixing takes place. This is temperature-controlled gas, and the sample is exhausted here. So typically, we like to work on a few seconds time frame delays at those temperatures. Again, I can tell you a lot more with the practical examples in the Raman practical room. So we've done that, and again, we're doing oxygen isotope substitution, 16-18 oxygen. We've done it across the different delay times. Notice we're looking at the 100 millisecond delay times, mixing those honey streams, right? So we're typically working pretty high pressures. And we can do that with the H-taurine and D-taurine. With deuterated taurine, we see a much slower reaction, which makes sense based on the kinetic isotope effect identified by the Penn State group. And we see a couple of species. Notice, I show you the Raman spectra in the gray. I kind of de-emphasize that. Don't look at that noise. Look at the nice, sharp, solid lines. Right? So that's one of the dirty tricks that we play. So how do I get them? Do I sit down and draw them by hand? Well, I probably should be fired if I were to do that. So we use modeling of Raman shifts. And if we're talking about two isotopomers, 18 oxygen and 16 oxygen, you can describe them as approximate by a Gaussian distribution of two modes. It's debatable whether you should Gaussian or anything else. Essentially, it's not important for our case. So any particular contribution of a particular spectrum D, which is frequency dependent, is given by this expression here. If you have single shift, great, you can do that, right? You simply fit it in any program that will handle the data. If you have multiple shifts, you will have a superposition of those shifts, and if they're well separated, it's good. But if they're overlapped, you cannot, your system is under constraint. You don't have enough information to understand where exact frequencies are. So what we're doing instead, we're using a global spectral regression where we acquire multiple spectra, and each of these spectra will have a frequency-dependent contribution of multiple species. And naturally, the frequency of the vibration will not change with the population. So properties like frequencies, width, and so on and so forth will be consistent across the entire data set, but each spectrum will have different and varying contribution of species A and B. It essentially increases the sensitivity of our method quite a bit. So, and we can do that. Notice here we have the same data set, and we have two scenarios where we have a single shift only, or two shifts, and you can see that once we get to the longer delay times, there is a clear discrepancy between a single shift and a double shift. That is an illustration. You can pull out very weak signals with this high degree of confidence if you do this kind of analysis, and you're obtaining the time-dependent profiles of all of the species. There are two possible scenarios with one shift or two shifts. So based on that, you can already say that the very last species here is the kind of afterthought in the reaction, and this is likely to be the first step in the process. So once we know what the frequencies are and we know how to model them, we want to ask the question, what's the chemical nature? And in this case, we'll be using mixed oxygen isotope again. So these are time-resolved spectra taken with the symmetrical isotopes and the asymmetrical isotope. You can clearly see that the double difference is giving you zero line. It means that in none of the species that we're observing, there are two oxygen atoms at the same time. All of the components, all of the species contain single oxygen atoms, meaning that the OO bond is already cleaved. That's an example from the first slide I have shown you. So based on that, we can invalidate all of these possibilities, which will fall in the right frequency range, but they contain two oxygen atoms. They cannot be contributing to what we're seeing in tau D, and therefore we can assign the frequency to iron 4 oxo vibration. And we do it again based on the frequency, magnitude of the shift, and the composition, isotopic composition of the species we're looking at. We can deuterate that, and when we first show those data, Some of colleagues over here took a long look at me. Are you serious about two-wave number shift with the signal-to-noise ratio like that? But remember, it's not simply eyeballing and picking up the right frequency, right? So this result of the calculation that fits all the data sets at the same time. So it is very constrained set. So what does it mean? Notice that we have an upshift only with the detaurine, but not necessarily dependent on protonation of bulk water. And that phenomena is actually well known for heme proteins, where if there is a hydrogen bonding between the terminal oxa and the base in the active site, then substitution of hydrogen for deuterium will shorten this bond, lengthening this bond, and decreases the localization of electron density into the hydrogen bond. So in hydrogen bonded system, in D2O, frequency goes up. And the magnitude of the shift varies. It's typically about few wave numbers up to 5 wave numbers with actually quite strong hydrogen bonding. So in this case, we do make that assignment based on this interpretation. Maybe even more interesting phenomenon happening with the low frequency mode here, where no matter what we substitute, there is no deuterium isotope sensitivity. So how do we interpret that? Based on the position of the reaction sequence, we think it is an RN3. But the RN3, something like that, analogy of the heme protein, so P450, should be proton sensitive, deuteration sensitive, and it is not. So we can clearly say, based on our results, it is not that geometry. Now, whether it is this geometry or that geometry is debatable. We are working on that. I'll leave it beyond the scope of today's talk, but at least there is precedent how this lack of sensitivity is explained. When the RNO stretch, the RNOH bond is acute, then the mass of the proton does not contribute to the vibration of RNO stretch, right? So under those conditions, this replacement of the isotope will not affect the bond much. Plus, it can be balanced by the hydrogen bonding, which have smaller but the opposite effect. We do not agree with that. We have data that suggests that even in the heme systems where that was invoked, there is probably another explanation for this, but again, I'm not going into that in so much details. And here is an example from Julieta Smolievich's work back in 1994 on host-radish peroxidase, where there is a very small upshift in deuteration. So that upshift in deuteration is then attributed to effect of this bond and the acute angle. We have now, as I said, data that suggests that there is no proton in this case altogether. So we can put together some sort of mechanism based on what we have seen. We observe RN4, so we're very comfortable with that. We propose that is controversial, so far we stand by that. It is an RN3, deprotonated RN3 oxo with the pK significantly lower than the model compounds, which then forms a alkoxo species, which I didn't have time to go into details today. There are two other explanations to that, and one of them is Sandevice did calculation on the reaction profile of tau D, and his calculations suggested two different pathways within about 7 kcal per mole. To the extent that you believe the accuracy of the number, and if you can argue that possibly there are changes, there are parallel pathways, it is conceivable that the two species that we're observing are appearing from different pathways. And the way to resolve that is to look at the early phase of the reaction. If these species, what we think is these, will appear concurrently with our ferryle, they will have to represent two parallel pathways without the crossover. And if it is sequential to initial species, then the sequence is clear, and then we have to deal with the PKs of that. And very recently, my grad student working on the FTIR, redox electrochemistry, of tau D, got a result that suggests there is actually significant perturbation in the active site with the redox change. So what we think may be happening is that following the oxidation, there is a reorganization of the ligand in the active site, which triggers the protonation. But again, that's beyond specifics of today's talk. What I'm showing you are the limitations of the Raman spectroscopy, what we can and cannot do. We cannot answer everything. And some things remain debatable. Okay, so that's for the mononuclear systems. How about dinuclear systems? And this audience does not need introduction for something like MMO. And here is the reaction of MMO. There are a few intermediates, and certainly compound Q will be of major interest for us. And as with tau D, we're going to ask the question, what is the absorption, what's the time domain? Where can we look at that? And luckily, stop flow data show that compound Q of interest here does have a prominent UV absorption. So we can excite it. And so what about the time domain? We'll be using, again, continuous flow approach. This is the lifetime of Q in the absence of the substrates, and it is giving us a very comfortable window of few seconds to look at our intermediate. So we can do Raman, transient Raman at those conditions. In this case, we really need to verify, considering reactivity of compound Q, we need to verify that we're really getting compound Q. So we have a built-in optical probe that will allow us to take spectra in the Raman instrument under the same conditions at the same point as the Raman measurements would be. And here is the spectrum. It is quite crappy, but it is not designed for optical measurements. There are much better instruments for that. It's designed for Raman, and this is our comparison to show that we are generating transient species with a spectrum which is consistent with established compound Q. We also have a luxury of using substrate, so we can add methane. In the case of tau D, reaction will not happen without the substrate, but here it will be, and we can look at the temporal changes in the populations to make an assignment of what we actually see. So we measure the 16 oxygen here. We're very excited. Look, there's a lot of stuff going on, right? A lot of vibration. This is the buffer. Well, bummer, some of the bands are gone, right? So, but we still have some bands. Well, I'll break it to you that most of those bands are actually plasma lines. None of them are really protein vibrations. So we have to identify the vibrations based on oxygen isotope. We do the 18 oxygen reaction. We compare them, and we see not much. Right? So we can take the difference. The difference will confirm that. We're not seeing much. Right? And it is only when you amplify the difference 25-fold, you begin to see the vibrations. So that's an illustration that you don't need to worry about the absolute spectra. You really want to look at the isotope difference Raman spectra if you want to make sense out of it. And in this case, we use the same approach as we use for tau D, except we could not use ethylene glycol. It is a concern to be a substrate for MMO. It will oxidize anything it will get on the active side. We had to do it close to freezing temperature of a natural buffer, but we had to make the system a lot more sophisticated, including different isotopes, and automate the whole process because it came to the point where human being could not sustain those measurements. But now we have an automated system, and we do the measurements in a sequence like that. This is the timing of our measurements, and in this particular case, 16-18 difference. It is done automatically. We can just watch. Then we'll separate it into different isotopes. We'll integrate them into the spectra that you saw on the previous slide, and then we calculate the difference that you have seen. So the success rate of this approach is somewhere about 90%. It's working. Surprisingly, to my astonishment, it does work. So here we have Raman spectra of MMO, no substrate spectrum. We have two oxygen vibrations, 1 and 2. Are they coming from the same species or are they separate? And what are the structures? So here we use the methane trick, and you can see that once you add the methane, this vibration is gone and this vibration is intensified. We've done it both methane and furan as an option. So it shows that this is catalytically competent, and it shows there are two distinct species that we're looking at. We'll have to make separate assignments for them. So we can actually add deuterated methane, and in that case, the lifetime is much longer. So we can now see at the QS complex where methane is bound to the active side, but the reaction hasn't happened yet. And we've done that, and what we're seeing that there is no vibration effect whatsoever. We'll come back to that in a second. There is some kinetic effect. It's decaying a little bit slower. So we recognize different species. We want to make an assignment of those species. And there are several possibilities. For the success of Q, the most likely scenario would be this. It has been shown that the methanol is released. One of the oxygen goes with the methanol. So it is possible that second oxygen remains in the cluster. However, for Q, there are multiple different scenarios. And it includes a closed core structure incorporating both oxygens, closed core with the one oxygen exchange, and an open core. As you know, you're not strangers to that. There is a debate whether one or another is the most catalytically competent. So we have to play some statistics here, right? So these are the different scenarios, different structures that we may observe in MMO. And let's first of all start with the symmetrical isotopes. If we start with the 1602 and 1802, in this case, we can have two different frequencies. In fact, in all of these cases, we can have two different frequencies. Notice, in the case when one of the oxygens is exchanged, the magnitude of the shift is smaller. This magnitude shift is larger, this magnitude is smaller. But it's difficult to decipher them primarily on the amplitudes of shifts, as illustrated for other cases. Now we bring in our heavy lifter, 1618 oxygen. And of course, these can bind with either one or another orientation to the active site. So we can have two equiprobable isotopomers. If we are dealing with a single oxygen bridge, naturally one of the oxygens will be gone, and depending which way it bound, it will be one or another. So it behaves like a ferrile in the first illustration I showed you. However, and the isotopic difference will be like that, absolute spectrum. In this case, it will be a little more interesting. Again, the same two frequencies, here and here. And it is only in the case if both oxygens are retained in the active site, we will have a new frequency appearing. Notice the amplitudes, right? 50-50 probability, amplitudes reduced in half. Now, knowing that, we can now go for the different spectra, and these, in this case, will have a different spectrum that looks like this. That's for the symmetrical isotopes. They all look more or less the same. But for the asymmetrical isotope, here we'll have a flat line. Here we'll have a flat line because we can have only one or another configuration. There is no two oxygen atom present. And only in the case of both oxygens in the active site will have intermediate frequency present. So those are the results, and you can see, make judgment for yourself. We believe that for T, it is clearly a single oxygen atom. There is no intermediate frequencies. And for Q, a new frequency is appearing that we are now detecting. So that tells us that this is the structure of Q, retaining both oxygen atoms. This is the structure of T, and these structures are invalidated. So that was very exciting. There is no isotope scrambling. It is also telling us about the mechanism of the O-bond cleavage. We're not observing that, but we can reverse engineer what the mechanism would be to have no scrambling in this case. And there are a few more questions, so we can do the iteration. I probably will going to skip that because there is something else I would like to show you, which is a lot newer just in the last few minutes. So for MMO, we have two possible mechanisms. One of them is heterolytic cleavage, which is shown here. Another one is a homolytic cleavage, and we show that MMOQ follows that pathway. There are related enzymes like ribonucleotide reductase, class 1A, class 1C, and the ADO, which again has been shown by the Penn State group to have very close structural similarity to class 1C RNA. So can we look at something like this, and can we decipher the structure of that? And for ADO, I'll skip a lot of information, just the basics. So it is the enzyme that will take aldehydes. It will use molecular oxygen electrons, releasing water, decarboxylated with the release of formate. So there is a very intriguing chemistry proposed for this enzyme. So the question is, what can Raman do for you? Can we look and can we identify any of these intermediates? This is actually the slide that I borrowed from Karsten, unchanged, I believe. Where he introduced different approaches taken to study this particular enzyme. And there is a lot of very useful information on that slide, but one thing I want to draw your attention to is this. Freeze quench and Raman. Freeze quench is a valid technique, but when it comes to Raman, because of photochemistry, it is very difficult to measure spectra of quenched intermediates that will be subjected to the photolysis, right? So it may be not the best technique. So maybe that's the reason why there were no success stories on ADO or some other major systems RFQ Raman studies. So what do we know about that? Based on the stop flow measurements, we know that the enzyme will bind the substrate. It will populate some species with the absorption in the visible, and then there will be a product of the reaction formed. When there is no substrate, the product is formed directly. So it happens something like that. If you have the substrate, we'll take the long route, and if you don't have the substrate, you will directly revert to that. We have the frequency window to examine either in the presence of substrate or in the absence of substrate, and we have the time frame, time domain, where we can study that reaction. So we can do the same thing as we've done with MMO and tau D, right? Well, maybe not so fast, especially for the photosensitive compound, and we know that ADO, peroxy intermediate, proposed intermediate, is photosensitive. And here is the reason. In our continuous flow method, the exposure of the molecule is defined by the distance between reaction start and the sample point and the size of the beam. Without going through a lot of details, our expected exposure for ADO for the conditions where we're doing that is roughly from 200 milliseconds to 1 second. That is how long it will take for molecule to travel through the laser beam before it leaves and is replaced with another molecule. Well, you may think that's a lot of time, right? In a focused laser beam, a lot of power. There is another method which is called spinning cell. And a spinning cell, it is a disk where you have a thin layer of sample rotating at a high velocity. And in this case, on the practical rates, the exposure of a time of single exposure is very short, 10 to the minus fifth. So the sample resides on the beam for a very short period of time. It has time to cool off before it is coming back and get measured again. So the equivalent exposure to the continuous flow method will be about 10 minutes of spinning cell for 100 microliter sample. So we decided, instead of going that route, we decided to initially examine that with the spinning cell. We had to have a temperature control, so we had to build a new instrument, and this is a departure from classical spinning cell Raman measurements. This is a cryogenic spinning cell setup. The spinning cell is right there in the middle, and there is a blow up. So this is the disk, the quartz disk. The beam is coming from the bottom. Raman scattering is collected at 90 degrees. In this case, we've done measurement at 0 degrees centigrade, and that difference is significant. But in principle, we have tested that system down to about minus 60, minus 70 degrees centigrade. So we can work with very cold systems using conventional and very economical route. So what do? We start with the O2 buffer. We start with the reduced ADO. We mix them together, open the vial, and we have a few seconds to do something with that. I mean, all of that is done manually. No automation, very low key. So we put it in a conventional spectrometer, Hewlett-Packard photodiode array, and that's what we measure. So by doing so, we can see that we are still generating, we can catch the onset of the reaction followed by the decay, and this is our optical window. It's going up and then it's going down. Now we can do the same, but now we can use 16 oxygen and 18 oxygen in our sample buffers. And instead of loading the sample in a conventional spectrometer, we put it in a spinning cell, and we measure one isotope, and then we measure another isotope. And if we look at the isotopic difference at the end of the reaction, there is not much going on. As we go to a shorter period of time, again, nothing. That was frustrating. Even here, we don't see nothing. And only at the very, very beginning, looks like we're having a squiggle of some sort in our reaction, right? So by the way, these measurements are repeated typically dozens of times. It's not a single measurement, right? So those dozens of measurements and different time frames are average. These spectra and these spectra are taken on the same sample or sets of samples. So what are those? And we decided to look at that a little bit more closely. And in this case, we are using two different measurements, and we're not looking at isotopic difference. We're looking at a temporal difference. So this is the time profile of ADO in a spectrophotometer, not the Raman system. We integrate different time frames, and then we use the very last time frame as our reference. We basically subtract this integrated spectrum from everything else. We normalize that to the Raman intensity, so the intensities here are meaningful. And now you're beginning to see that in the first spectrum, you have a number of vibrations. The trained eye will actually pick that there may be some isotopic difference. Notice that the red is peaking a little bit more at these two locations from the blue. Then that mode is gradually disappearing, and by the time we get here to 100 to 200 seconds, it's pretty much gone. That was taken at 50 milliwatt, and remember, we have to ask the question, is it real or is it a photo product? And we've done the same measurement at 90 milliwatt with the thought that if whatever we are seeing here is a photo product, the amplitude or intensity of that will go up, right? Or if it is photosensitive, then maybe the intensity will go down, or maybe something else. Notice that, again, they're normalized in exactly the same way. At 90 milliwatt and at 50 milliwatt, the spectra are essentially identical within the noise consideration that the disclaimer that I gave you. And you can see the isotopic difference a little bit more clearly between them. What is interesting that in 90 milliwatt measurements, you can see that only in the first spectrum. Essentially, all the features are gone by the second time frame, while in 50 milliwatt, they're persisting for a longer time. So what we're seeing is not that we're getting less of the compound, but it decays faster, higher laser power. So you also can see, no, that's basically what is going on here. You also can see that the power has a major role in the signal-to-noise ratio. If you cut the power in half, you have to quadruple your accumulation time to make up for that. So that's why we really like to stay the higher power if we can do so. So based on that, we can now calculate isotopic differences. And again, you see the same squiggle as you saw in the first spectrum. In fact, you can see one squiggle here and another squiggle there. And the squiggle is gone as those absolute features are disappearing from our time-resolved spectrum. So this is a real-time, time-resolved isotope difference spectrum of ADO. That's what we're seeing. And how can we interpret that? Great. Maybe we have the peroxy bond. Does anybody see a problem with the values that are here? What's the magnitude of the isotopic shift? It's almost 100 wave numbers. Right? So if we go in the literature and we look what should be the magnitude of isotopic downshift, it's somewhere about maybe 50, right, on a sunny day, maybe even less. So these cannot come from the peroxy intermediate. In fact, it cannot come from any structure, single structure. So what probably is happening, we have a pair of isotopic shifts present simultaneously where the 16-oxygen isotopomorph of this mode is canceling the 18-oxygen isotopomorph of another. And if that's the case, then the possible assignment for that would be the higher frequency mode may be coming from the OO stretch. What will be the lower frequency one? The likeliest scenario in that case would be either a equilibrium between two peroxy species or possibly an CO stretch, which will also come in about the same region. We have done modeling for other systems, and it is not unexpected. Lastly, the low frequency feature may very well come from the bridging mode here, and from the same intermediate or maybe a product of that. I don't have time to go through all details. We've done studies on the dipheric ADO. I'm going to skip that. There is unfortunately a lot of interesting things. You'll see that in the uploaded version of that. And there are some illustrations for deuterium sensitivity. There are two different dipheric ADOs, even though our colleagues to my left adamantly state that there is only a single Moss-Bauer visible sample. So we'll strongly disagree, and here is the evidence for that. But we can work it out in a regular fashion. In our interpretation, we have those intermediates that we're probably detecting. We cannot unambiguously say whether it is this structure or this yet, but we have the signature to look at. That information is coming from the other information, not from our studies directly. We haven't looked at that formation stage. This is the mechanism that has been proposed for the enzyme by the Penn State group. And then it is completed with the release. What we are seeing, however, that there are two distinctly different states, not only vibrationally, but also in terms of the hydrogen and deuterium sensitivity, which are photosensitive, and there is a clear conversion of one into another. And so these are exchangeable with the bulk water. These are not exchangeable coming only from the O2 molecular oxygen. And with that, all I can do is to thank those who were involved in that. And Pyotr Grzishko has done a fantastic job on the Tau-D studies and a very dedicated person. Raul was driving the MMO work. The ADO work that I showed Raman was done by Yegor and our colleagues at Penn State, and particularly Maria now, in independent position, provided the sample. By the way, to give you a reference about the amounts, John Lipscomb will love to tell you that he provided 15 grams of purified protein for our ADO work. I really would like to redeem myself to say that the isotope difference spectra of ADO took about 3 micromoles of protein, which is much more reasonable quantity. And that's how typically it goes. And again, the question I'll pose to you is not what you can do for the Raman, but what the Raman can do for you. By all means, we're interested in hearing from you. in the systems that may have answers in that approach. Thank you.
We have time for one quick question because all the teachers need to go prepare.
I have 5.
Okay.
That means 4 videos.
That right, six-pack, yes. I would go for six-pack.
First of all, I don't know how we would say there's only one ferric species. The only two we have today is Moscow, and we know how poor the resolution is.
Right.
So we can't say that, so we totally accept it.
Okay. Well, it would be very interesting to see if there are correlation between the Moss-Bauer signatures and
But one other thing, I struggle with the ferric state in calculus. The reason is this: the isotope effects on the decay of the feral state are huge.
right.
You can't really have any kinetically significant step between feral hydrogen abstraction and ferrous forming.
Right.
And if you had some ferric accumulation, then when you slow that species accumulation by the isotope effect, you get much less.
I absolutely agree. So what?
You could have a second isotope effect, maybe on removal of the proton, which
Right.
is exactly the same
Right.
as the initial.
Right.
It may
Yeah,
be
I don't think that will be practical. You're absolutely right. It is clearly hydrogen atom transfer. So in all the mechanisms that we proposed, the very first step keeps the proton on the oxygen. The question is not whether it is abstracted or not. The question is why would the species not retain that proton after the hydrogen atom abstraction? But the Bordwell cycle in terms of the dynamics of that is exactly right as you say.